Okay, so sorry for the delay. Okay, so we are going to continue on chapter 7. Okay, so on our last lecture, we have gone through basically the overview of the immune system. You know that the immune system has three line of defense. Okay, three line of defense, which is you have the external barrier, the internal barrier, and also the acquired immunity, the third line defense. Okay, the acquired immunity. So the first and the second line of defense, it protects your body against whatever pathogen that tries to enter into your body. And as for the third line defense, which is the acquired immunity, it protects your body against specific pathogens. Okay, specific pathogens. So okay, so let's look at this diagram. I've gone through this diagram also, where the immune system, the animal immune system, it consists of the three line defense. Okay, which is the innate immunity. Okay, so saya Okay, so the innate immunity, which recognize the trait shared by broad range of pathogens using a small set of receptor. So innate immunity. So innate immunity, it is divided into two, which is the barrier defense and also the internal defense. The barrier defense is the first line defense. The internal defense is the second line defense. Okay, and then the adaptive immunity is the third line defense. So for the first line and second line defense, it recognize the feature of the innate immunity is that it protects your body against the invasion of many pathogens. So whatever pathogen that tries to enter into your body, that is the function of the innate immunity, to protect your body against the invasion of these pathogens. So recognition of traits shared by broad ranges of pathogens using a small set of receptor, and the response is rapid. So it is there. Your skin is there to protect your body from the entrance of pathogen, for example. Okay, the skin. And then the mucous membranes, and then also the secretions such as tears. Okay, tears also protects the entrance of pathogen through the eyes, for example. Okay, and then internal defense is the second line defense, which involve phagocytic cells. So there are cells that will engulf, okay, whatever pathogen that it encounters. And then natural killer cell, it destroys, okay, this cell, we call it as NK cell. It destroys infected body cells, okay, infected body cells, and exposing pathogen in the infected body cells. Okay, it exposes the pathogen, for example, towards antibody that is circulating in your body fluid, or it exposes the pathogen to macrophage, so that macrophage can engulf the pathogen. So that is the function of the natural killer cell. It destroys infected body cells. And then you have the antimicrobial protein. So this one, we are going to look at the function of the antimicrobial protein. Basically, it involves, for example, a group of protein that will embed in the plasma membrane of the pathogen, and causes there's mechanism that causes the pathogen to be destroyed. Okay, and then inflammatory response. We are going to look at the mechanism later. And then for the third line, the adaptive immunity, recognition of trait specific. So you have to highlight this. Specific to particular pathogen using vast array of receptors. And the response is slower. Okay, so it involves a series of reactions to occur. For example, you have macrophage that encounters the pathogen, and then the pathogen will be digested by the macrophage into fragments, and then this fragment will be exposed, okay, to the surface of the pathogen, and there's another cell that has receptor for that that matches with the antigen, okay, with the antigen presented by the macrophage. The macrophage, then we call it as APC. So the story is quite long, so that's why the response is slower until it produces at the end, for example, the production of antibody against that specific pathogen. Okay, so that's why it says here for the adaptive immunity, it is divided into two. Okay, which is the antibody mediated immunity or humoral immune response. So it involves antibody circulating in your body fluids, okay, circulating in your body fluids to find any pathogen that are in your body fluids. So body fluids involves the blood, your blood in the blood vessels, or in the interstitial fluid, the fluid surrounding your body tissue, and also the lymph. Okay, so these are your body fluids. So the pathogen can be found anywhere in your body fluids. So you also have antibody in the in your body fluid that function to find that specific pathogen, so that the antibody can bind to that specific pathogen. So you have to remember, the antibody will not destroy the pathogen directly. Okay, that is not the function of antibody. It does not destroy the pathogen directly. So there are some mechanisms that we are going to look at later on how the antibody works. Okay, and then that is the humoral immune response, okay, or the antibody mediated immune response. This one involves the B cell, okay, B cell, B lymphocyte, okay, B cells. And then the second type of adaptive immunity is cell-mediated immune response. Okay, so for this one, it involves the cytotoxic T cell. Okay, cytotoxic T cell or cytotoxic cells, it says here. So it defends against infection in body cells. Okay, infection in body cell. So the mechanism posed by the cytotoxic cells is similar to NK. Except that the cytotoxic T cell it destroys specific infected body cell against specific pathogen. Okay, so any if the host cell is infected by that specific pathogen, so that is the function of the cytotoxic T cell, to destroy that cells, to destroy host cell infected by the pathogen and exposes the pathogen. Okay, so that is the function of the cytotoxic T cell. It has similar function to NK, but NK is in the second line defense, in the internal defense. Cytotoxic T cell is in the third line defense. Okay, so let's look at innate immunity first. It is non-specific, so this one you have to remember. So this is the feature of innate immunity, non-specific defense system. Why? Because it detects a broad range of pathogen using a small group of receptor proteins, and the response is rapid. Okay, the response is rapid. It is there to protect your body against the invasion of pathogen, your skin, for example, right? And then some response to pathogen, same response to pathogen each time. Mechanism tu sama lah kan. Your skin is there to protect your body against the entrance of pathogen. The tears that you produce, okay, to wash off or to provide a washing action, okay, against the entrance of pathogen, or mata awak masuk abuk contohnya kan. So you produce tears, okay. So that is another example. So the response is the same each time, okay, and then found in all animals. Okay, so external barrier. So ingat ya, so this is the first line defense. Okay, the first line defense. So for example, you have here lysosome. So tadi saya bagi example tears kan, air mata. So in the tears it contains lysozyme. Okay, lysozyme that will degrade the pathogen, for example, that tries to enter. So you have other secretion also that serves to protect the entrance of the pathogen. And then you have removal of particle by cilia in the nasopharynx. Okay, nasopharynx. So dalam rongga hidung awak kat dalam ni, so they are cilia. Okay, that serves to trap the pathogen from entering deep into your respiratory system, into your trachea, for example, and prevent from the entrance of the pathogen into the lungs. Okay, and then the trachea also have mucous lining. Okay, mucous lining. So the lining produces mucus to trap the pathogen that tries to enter into the lungs with the air that you breathe in. So the air that you breathe in, obviously it contains many contaminants, many pollutants. Okay, and then in the stomach, so you have hydrochloric acid, can HCl. So this gives an acidic environment in the stomach. Okay, in the stomach, so that when you eat food that are not clean, it is exposed to bacteria or pathogen. So once the food is inside the stomach, so these acids will also destroy the pathogen along with the food that you eat. Okay, and then you have a rapid change of pH. So in the stomach is acidic, but once the food enters into the intestine, it is alkaline, for example. Okay, and then you have the skin. Skin surface provides a physical barrier. So it has fatty acids and also normal flora. Okay, normal flora is the natural bacteria that's on your skin. Okay, so this prevents the growth of the bad bacteria, for example. And then also you the same in the intestine, in the large intestine. So you have the normal flora. Okay, and then the flushing of urinary tract. Okay, so this will wash off any bacteria or pathogen that is in the urinary tract. Okay, so the barrier defense will include the skin, okay, mucous membrane, and then secretion from the skin and also the mucous membranes. Okay, so let's look at the skin first. So the skin is an impenetrable barrier. Okay, it is home to normal flora, non-pathogenic bacteria. Okay, so that prevents the growth of pathogens. So when you have normal flora, so this prevents the growth of the bad guys, the pathogens. Okay, so so this is the a diagram, okay, showing you the structure of the skin. So the top layer you have the epidermis, and then you have the dermis, below the epidermis, and then you have the hair follicles, and then you have here the oil glands, okay, the sweat glands. Okay, so those are the structures that are in the skins. Okay, so the sweat produced, okay, will also serve as the to wash off, okay, any pathogens that are found in your skin. Okay, so and then you have the mucous membrane. Okay, so the mucous membranes are made up of epithelial cells that line the digestive, the respiratory, and urinary, and also the reproductive tract. Okay, so these specialized cells that are scattered between the epithelial cells secrete mucus that trap pathogens. Okay, so the mucous membranes that are made up of epithelial cells can secrete mucus that function to trap any pathogen. Okay, that enters, okay, into your digestive system along with the food that you eat, or through the air that you breathe in the respiratory system, or in your urinary tract, and also in the reproductive tract. Okay. So the pathogens are eliminated by specific mechanisms. Okay. For example, let's look at the mucous membrane that lines the digestive tract. Okay, digestive tract. So pathogen enters the stomach with the food that you eat. Okay, so the food that you eat are exposed to pathogen. For example, it is not clean, for example, and then the pathogen are destroyed by the stomach acid. Okay, the HCl, hydrochloric acid in the stomach, that gives an acidic environment of pH 2, for example, and then digestive enzyme in the intestine. So the pathogen in the stomach, along with the food that you eat, will be digested by pepsin, and then the stomach acid, and then after that it becomes the food that then enters into the intestine, and then encounters digestive enzyme. So this digestive enzyme can come from the pancreas. Okay, so pancreas secretes digestive enzyme into the small intestine. Okay, and then the small intestine also produces its own digestive enzyme. So all this enzyme will digest the food along with the pathogen. Okay, that may be present. Okay, and then normal flora in the gastrointestinal tract. So the normal flora are the good bacteria that prevents the growth or the invasion of the pathogen. Okay. And then next is the mucous membrane that lines the respiratory tract. Okay, the respiratory tract starts from the nasal cavity, the pharynx, the larynx, the trachea, before it enters into the lungs. Okay, that is the respiratory tracts. So the epithelial cells that lines the respiratory tract also have cilia. So that is the feature of the respiratory tract that starts from the nasal cavity, the nasopharynx area, kan? So sebelum dia masuk kepada trachea tu, so it has cilia. So the cilia traps the pathogen. So pathogen present in the inhaled air are trapped by the mucus within the trachea, bronchi, and also bronchioles. The cilia will swipe the mucus upward, upward the glottis, to be swallowed in the digestive tract. Okay, so if you were to observe this diagram, kalau dia kan, so kat sini akan ada junction. This is the esophagus, kan, esophagus, and then this depan esophagus, depan ni ada trachea, kan, belakang trachea ada esophagus. So lining here you have the cilia. Okay, lining the nasal cavity, the nasopharynx, you have the cilia, and then area kat sini, if I'm not mistaken, is the glottis. Kan? So you have cilia, you have the mucus trapping the pathogen. So the cilia will swipe the mucus containing the pathogen upward, until it enters into the esophagus. Kat sini ada cilia semua, kan, juga. It produces mucus, so dia akan swipe upward. The cilia will swipe upward the mucus containing pathogen until it is swallowed and enters into the esophagus. Okay, and then in the esophagus, then it will be destroyed in the stomach by the stomach acid. Okay. Nampak ni, specialized cell that are scattered between epithelial cells secrete mucus that trap pathogen. Pathogen are eliminated. Inilah, okay. Cilia ni. The cilia swipe the mucus upward towards the glottis to be swallowed into the digestive tract, enters into the esophagus, and then enters into the stomach to be destroyed by the stomach acid. So next is the mucous membrane that lines the respiratory tract. Sama je, okay. Cilia. And then you have a specialized cell, we call it as the goblet cell. The function of the goblet cell is to produce mucus. Okay, goblet cells is to produce mucus, and the structure of cells that line the respiratory tract, okay, especially at the trachea region, the structure of the cell is pseudo-stratified epithelial cell. Kita belajar dulu kan masa chapter 2 tak silap saya semester lepas. Okay, the different structure of cells kan. So kalau untuk pseudo-stratified ni is cells that is single layer but the cell of varying height or length kan. So it seems that the layer has the tissue layer lining the trachea has many layers, but it is actually one layer of cell due to the different length of the cell. Okay, ada yang kecil, ada yang panjang, macam tu kan. So it is also has cilia. Alright, it also has cilia. Cilia and also the goblet to produce mucus. So mucus produced will trap the pathogen, and then the cilia will swipe upward until it reaches the junction between the esophagus and also between the trachea to be swallowed into the esophagus. Okay. So next is the mucous membrane that lines the female reproductive tract. You have the for the female is the vagina, so that secretes a sticky and acidic secretion that kills pathogen. Okay, so promotes the growth of normal flora that prevents the growth of pathogen. So that's why when we look at chapter on reproductive system, the male accessory gland will produce alkaline fluids. Okay, so this is to neutralize the acidic environment of the vagina. So the vagina features or characteristics is that produces acidic secretion that to kill off the pathogen. Okay, and it also has normal flora. Okay, that prevents the growth of pathogen. So next is the mucous membrane that lines the urinary tract. Okay, so for both male and female. So secretion from the epithelial cells such as antibacterial agent that inhibits bacterial growth. Okay, so the epithelial cells produces and secretes antibacterial agents, okay, that inhibit bacterial growth. So the urine is also acidic. Okay, so acidic which inhibits also the growth of bacteria, and then the urine cleanses the lower urinary tract. The urine provides a flushing action. Okay, so it flushes away the pathogen in the urinary tract. Okay, so it is acidic. It provides a washing action, a flushing action. It flushes away the pathogen from the urinary tract. Okay, so ada tiga feature ni lah. Antibacterial agent, the urine is acidic, and also provides a washing action to cleanse the urinary tract of any pathogen. And then next is the secretion from the skin and the mucous membrane. Okay, secretion from the skin and the mucous membrane. So the skin produces sweat, for example. Okay, produces mucus, and then saliva and also tears. So these are hostile to many microbes. Okay, so oil and sweat glands give human an acidic pH. Okay, acidic environment of pH 3 to 5. Okay, so the acidic environment will inhibit the growth of pathogen. Okay, so it contains lysozyme that degrades bacteria cell wall. Okay, so pernah ada macam soalan applied. Okay, for example, dia boleh tanya, a person applies lotion, okay, that of pH 7 or pH 8. Is it is it safe to be used? Is it on the skin? Macam tu lah kan. So your skin condition supposed to be acidic. Okay, due to the production of the oil and also sweat by the sweat gland. So this prevent the growth of pathogen. Okay, so those are the first line defense. Okay, the first line defense, the barrier defense, which involve the skin, the mucous membrane lining the digestive tract, respiratory tract, the urinary tract, the reproductive tract. Okay, so those are the mucous membrane, and the secretion from the mucous membrane, and also the skin, oil, and also sweat. Okay. So next we are going to look at the second line defense, or the internal defense. So if the pathogen manage to penetrate the first line defense, it will encounter the second line defense, which is the internal defense. Okay, so the internal defense may involve phagocytic cells, okay, such as macrophage. Initially it was monocytes, so the monocyte will mature into macrophage. And then you have neutrophil and also eosinophil. So these are the phagocytic cells that you have to know. Examples of phagocytic cells: white blood cells that can engulf pathogen. Okay, so you have to know that specific phagocytic cell. Sebab kalau lymphocyte, macam contohnya B cell or T cell, it cannot engulf pathogen, only certain white blood cell. Okay. Okay. So if you look at this diagram, you have all the different types of cells. Okay, obviously you have the red blood cells, but the red blood cells involves in carrying respiratory gas and also nutrients to remove waste. That is the function of red blood cells, the erythrocyte. You have another cell which is the white blood cell. So white blood cells, so there are many types. You have the monocyte, okay, that matures into macrophage, and then you have the neutrophil, and then you have the eosinophil. So these are the phagocytic cell. Okay, and then you have basophils, and then you have the lymphocyte. So lymphocyte ada T cell, ada B cell. T cell ada helper T cell, ada cytotoxic T cell. Okay, helper T cell and also cytotoxic T cell. So okay, the internal defense, it is also known as the second line of defense. Okay, internal defense. So it is triggered when the pathogen enters the body. It penetrates the first line defense. It penetrates the skin, for example. So the pathogen will encounter the internal defense, which involve all these four components. Okay, which is the phagocytic cell. Phagocytic cell tadilah neutrophil, macrophage, and also eosinophil. And then you have the antimicrobial proteins. We are going to look at the mechanism exhibited by the antimicrobial proteins. And then the NK cell, the natural killer cell, and also the inflammatory response. So all these you should know the explanation. Okay. So let's look at the phagocytic cell first. So this type of defense is known as the cellular counter-attack. This one you have to remember. Okay, cellular counter-attack, because the phagocytic cell or the phagocytes detect the pathogen and attack them. Okay, so nama kita boleh panggil dia sebagai phagocytic cell or phagocytes. Okay, so phagocyte will attack the pathogen by engulfing the pathogen through the process of phagocytosis. Okay, so the phagocyte kills the pathogen by engulfing them through phagocytosis. Okay, so phagocytes have receptors that recognize certain structures, such as polysaccharides on the bacterial cell walls. Okay, so the basic it means that the phagocytic cell will detect common molecules that are found in most bacteria, for example, okay, or in most of the bacterial cell wall. So the phagocyte then engulf the bacteria and trap them in vacuole. Okay, proses phagocytosis lah yang kita dah belajar dulu kan, semester 2 masa chapter 3, the process of phagocytosis. So the bacteria engulf, the phagocytic cell engulf the bacteria or the pathogen, okay, and then internalize, and then package into vacuole. Okay, the pathogen is inside the vacuole to be digested. Okay, digested by how? The vacuole fuses with lysosome. Okay, the lysosome contains lysozyme or hydrolytic enzymes. Okay, And it also produces nitric oxide, free radical, and other gases that poisons the pathogen. Yeah, so first the lysosome, the lysosome contains nitric acids and other gas that poisons the pathogen, and then the lysozyme inside the lysosome and other enzymes will degrade the bacteria cell wall. Okay, it destroys the bacteria. So as you can see from this diagram here, you have the pathogen outside, for example in your body fluid. Okay, so nanti datang phagocytic cell, macrophage ke, neutrophil ke, ataupun eosinophil ke datang, and then dia akan engulf by forming what? Pseudopodia first. Okay, pseudopodia cell extension, okay, that surrounds the pathogen, and then the pathogens are engulfed by the process of endocytosis. Sama lah, endocytosis adalah salah satu mekanisme phagocytosis. Okay, phagocytosis internalized. Okay, endocytosis, and then the phagocytic cell that has engulfed the pathogen will surround the pathogen inside a vacuole. So the vacuole then will fuse with lysosome. So the lysosome containing enzymes such as enzyme nitric oxide also, and then that mechanism will destroy the pathogen. It destroys the pathogen by degrading the pathogen into fragments, and then the fragments or the debris will be released by the process of exocytosis lah kat sini. So bawa masuk endocytosis, bawa keluar exocytosis. Okay, so engulfing is phagocytosis. Okay, so these are examples of phagocytic cells. You have to remember macrophage. Okay, initially macrophage ni dia adalah monocytes. Okay, and then you have neutrophils, engulf and destroys pathogen. The most abundant white blood cells is the neutrophils. When you are infected by pathogen, the most abundant is the neutrophil. Okay, so when you are infected by certain pathogen, yang akan datang dengan banyaknya adalah neutrophil. And then you have the eosinophil, limited role in phagocytosis. So maksudnya eosinophil, it can engulf the pathogen, but it has other function as well. The main function of another function of eosinophil is to destroy multicellular parasites. Okay, multicellular parasites that have many cells. Okay, so parasites. So obviously the eosinophil cannot engulf. So it has granules that has enzyme. So this enzyme will be secreted to destroy that multicellular parasite. So eosinophil dia boleh juga it can undergo phagocytosis to destroy pathogen that are unicellular, and it can also destroy multicellular parasites. Okay, by secreting enzymes that will destroy that multicellular parasites. Okay, so that's why dia cakap kat sini limited roles. So bukan main function dia, tapi dia juga boleh bukan main function eosinophil untuk engulf, tapi dia boleh juga engulf. Okay, okay, so the next one is dendritic cells. Okay, limited roles in phagocytosis. So if you look at these two cells, dia ada overlap. Okay, macrophage awak tengok kat sini, dia terlibat dalam second line defense. And once the pathogen is engulfed in the second line defense, it can trigger the third line defense to be activated. Okay, macrophage, once it engulfs the pathogen in the second line defense, any pathogen that it encounters, okay, so that mechanism will trigger the activation of the third line defense. As the macrophage convert into another cell, we call it as APC, antigen presenting cell. Okay, initially dia adalah monocyte matures into macrophage, engulf pathogen, okay, degrade the pathogen, and then the pathogen becomes into fragments, which is we call it as antigen. So this antigen can, so this is the macrophage. The antigen will be exposed on the surface of the macrophage cell, and this macrophage then will convert into another cell. We call it as APC, antigen presenting cell, in the third line defense. Okay, so later we are going to look at in details the mechanism in the third line defense by the macrophage. So that they are overlapped kat situ. Macrophage involved in both second line defense and also third line defense. Okay. Same goes to dendritic cells. Dendritic cells can also involve in both second and third line defense, if I'm not mistaken. Nanti kita tengok. Okay, dendritic cell. Second and third line defense. Okay, dia ada overlap. Tapi kalau neutrophil, eosinophil memang dalam second line defense sahaja. Okay. Okay, so the macrophage, large irregular shape of phagocytes known as big eaters. Okay, big eaters. So developed from monocyte in the blood, patrols the body tissue, engulfing damaged cells and foreign matter. Other resides permanently in organs and tissue. For example, lungs air sac, which is the alveolus. Okay, it also resides in your alveolus, in the lungs. Okay, to destroy foreign matters from the air that you breathe in. So the air that you breathe in may contain pathogens, really small virus, for example, that enters into the lungs. So this virus will or pathogen will encounter macrophage that is in your alveolus. Okay. So next is the dendritic cell. Okay, it roams roam in tissues that are in connect with the environment. So migrate to the lymph nodes after encounter the pathogen. Okay, so migrate to the lymph nodes after encounter pathogen. And in the lymph nodes, dendritic cells interact with immune cells to stimulate adaptive immunity. Nampak? Tadi kan saya tunjuk macrophage and also dendritic cells, dia ada overlap kat situ. Dia boleh involve in second line defense, engulf, okay, engulf, and then in the case of dendritic cell, it goes to the lymph nodes, and then these two cells will convert itself into a cell. We call it as antigen presenting cell. Okay, antigen presenting cell, where this cell, once it engulfs the pathogen, it degrades the pathogen into fragments. We call it as antigen. Exposed to the surface of this phagocytic cell. Sebab itulah kita panggil dia antigen presenting cell. It presents the antigen of the pathogen on the surface of the dendritic cell and also macrophage. It exposes kepada siapa? Kepada another cell, which is the helper T cell. Okay, the T helper cell. Dia bergantung kepada buku yang awak guna. Kalau buku Campbell, dia guna helper T cell. Tapi kalau awak guna buku lagi satu yang you all ada ni, Solomon Point ke apa, dia guna istilah ni, T helper cell. Okay, sama je, helper T cell, T helper cell. Saya dah terbiasa sebut helper T cell, tapi sebab kita revise you all punya notes according to the reference book that you use. Okay, so it uses the term T helper cell. So it is the same. Okay, T helper cell and activate the cytotoxic T cell. Okay, so helper T cell will bind to the antigen presented by the APC. The helper T cell activate another cell, which is the cytotoxic T cell, for example. So once the cytotoxic helper T cell activated, it goes to the infected host cell to destroy the infected host cell, for example. So the detailed mechanism we are going to look at in the third line defense. Okay, so next is the neutrophils. Okay, another types of phagocyte. As I said, it is the most abundant. Okay, so when you are infected by a virus, awak demam ke, ataupun you have a cut, for example. Okay, so the neutrophils is the cell that will straight away goes to that infected area. Dia yang akan pergi dulu kat infected area, dia yang datang pada jumlah yang banyak. Okay, it is the most abundant. Okay, abundant leukocytes. It circulates in the blood, attracted to the injured sites by signals from infected tissues. Okay, infected tissues will produce chemical molecules that will signal this neutrophil to come to that injured site, to the wound. For example, you have a cut or a wound, so the neutrophil will come to that wound because the injured tissue will produce signaling molecules that will attract the neutrophil to come to that area. And then, so once the neutrophil reaches to the wound area, you have a cut. For example, the cut is exposed, so it is so you have the a higher chance of pathogen to enter into that cut area. So then, the neutrophil will engulf any pathogen or bacteria before the bacteria manages to enter deep into your body. Okay, so phagocytose bacteria before inactivated. Okay, so next is the eosinophil. Can be found in mucous membrane that lines the digestive, respiratory, and also urinary tract. So this is where you can find the eosinophil. It lines the digestive, respiratory, and urinary tract. As I said, it defends against multicellular invaders, multicellular pathogens, multicellular parasites. Okay, parasites. So by what mechanism it position? The eosinophil will position themselves against the parasite body and destroy the parasites by secreting destructive enzymes. Okay, eosinophil ni dia boleh engulf pathogen dan dia juga boleh destroy multicellular parasite by secreting destructive enzyme to the parasite, to destroy the parasite's body. Okay, so those are the first type of the second line defense, which involve the phagocytic cell or the phagocytes. Ingat dia ada macrophage, ada dendritic cell, ada eosinophil, ada neutrophil. You have to remember examples of phagocytic cell. So next you have the second component of the second line defense is the antimicrobial proteins. Okay, antimicrobial proteins. So the antimicrobial protein consists of peptides and also proteins. Okay, so that's why we call it as antimicrobial protein. It contains of peptides and also protein that function to kill, prevent, or inhibit the growth or reproduction of pathogen. So examples of antimicrobial protein kita ada interferons and also complement system. So you have to know the mechanism by each of these component. Okay, so the interferon macam mana dia berfungsi, complement system macam mana dia berfungsi. Okay, so the interferons can. The first type of antimicrobial protein. So the function of the interferon is to so interferons are protein, okay, protein that inhibit viral reproduction. Okay, inhibit viral reproduction. So secreted by virus infected body cells. Okay, so the host cell that is infected by the virus. So virus is inside the host cell. So the host cell will produce infected host cell will produce the interferon. Okay, interferon. So this interferon is not for the cell itself. It goes to another cell that is not yet infected by the virus. Okay, so this stimulate uninfected cell, the healthy cells that are not infected yet by the virus, for example, to produce antiviral protein. Okay, so healthy cell, okay, the unaffected cell will produce the antiviral protein. So this protein will block the entrance of the virus in the healthy cell. Okay, that blocks the viral reproduction. So help control viral infections such as cold and also influenza by limiting cell-to-cell spread of the virus. Okay. Other type are interfering viruses, helping activate macrophage, these enhance their phagocytosis. Okay. Okay. So the interferons also activate macrophage, so that the macrophage can engulf the pathogen better. Okay, enhances the process of phagocytosis. So as you can see, so this is the host cell 1. So the host cell 1 is the host cell that is infected by the virus. The virus is inside the host cell. So it says, okay, as you can see, so this is the virus. Inside the virus, you have the RNA of the virus. Okay, so the viral nucleic acid, RNA. So the RNA will enter into the host cell, the infected host cell, and triggers a segment, a gene in the DNA of the host cell to be activated. Okay, interferon gene is activated. Okay, interferon gene is the gene inside the host cell. Host cell yang punya. Okay, host cell yang punya. So once this interferon gene is activated, it undergoes the same process of transcription, translation. Transcription produces mRNA. Translation produces the protein. Okay, translation produces the protein, which is the interferon molecule. Interferon molecule are protein, kan? Proteins. Okay, so this interferon molecule will be secreted out from the infected host cell. It finds another cell that is healthy, still healthy. It is not infected yet by the virus. Okay, so this interferon protein will enters into the healthy cell. Okay, the uninfected host cell. So it triggers the activation of gene inside the host cell that produces antiviral protein. Okay, antiviral protein sama lah. Dia akan once the gene is activated, produces mRNA through transcription, and then through translation produces the antiviral protein that blocks, okay, the reproduction of the virus inside the healthy cell. So this will limit cell to cell spread of virus. Okay, limit cell to cell spread of virus. So ingat ni ada dua. Interferon by the infected host cell, antiviral protein by the healthy cell. Okay, antiviral protein is by the healthy cell, by the uninfected host cell. Maybe awak boleh letak kat sini lah, nak bagi awak lagi mudah faham, kan? Uninfected host cell. Okay. You have here a video. Let me just save. Sekejap, type of antimicrobial protein. Yeah, the next type of antimicrobial protein is the complement system. So antimicrobial protein, the first one you have the interferon, and then second you have the complement system. So for the complement system is a group of approximately 30 proteins in the blood plasma. Okay, 30 different types of protein in the blood plasma. So it circulate in an inactive form. So circulate dalam keadaan yang tak aktif, and are activated by substance on the surface of pathogens. Okay, so the activation triggers the generation of membrane attack complex. So the pathogen, so on the surface it may have a molecule or antigen. Okay, so this molecule on the surface of the pathogen will activate. Okay, it activates the complement system, the protein of the complement system to trigger membrane attack complex. Okay, membrane attack complex. So the complex insert themselves into the plasma membrane of the pathogen and form a little hole. Okay, a little hole yang berbahaya lah, kan, pada plasma membrane of the pathogen. So ini pathogen. Okay, so you have the circulating protein. Okay, the circulating protein. So this circulating protein will be activated by the molecule on the surface of the pathogen, and this protein will be inserted in the plasma membrane of the pathogen to form a little holes. Okay, hole yang berbahaya. Kenapa berbahaya? Once the hole is formed, the pore is formed on on the surface membrane, on the surface of the plasma membrane of the pathogen. So ions and water can enter inside the pathogen and causes the pathogen lama-lama dia akan burst. Okay, dia akan lyse and then burst. Okay, so this is the complement protein. So complement protein it circulates in your body fluids, but once it encounters the pathogen, it insert the complement protein will insert themselves into the membrane of the pathogen to form holes or pores. Okay, so this causes fluids containing ions and water to enter inside the pathogen and causes the pathogen to burst. Okay, and lyse and then burst. Okay, swell and then burst. Burst and lyse is the same thing. Okay, swell and burst. Kita nak cakap perkataan swell tu tadi. Okay, swell and then burst. Dikembang dan dipecah. Okay. So extracellular fluid enters the pathogen through the pores, causing the pathogen to swell and then burst. Other complement protein coat the surface of the pathogen to enhance phagocytosis of the pathogen and by the macrophage. Okay, so other complement protein coats the surface of the pathogen to enhance phagocytosis. Okay, so how does the process enhances the phagocytosis? By binding to the surface of the pathogen, the complement system can inactivate the pathogen, for example. So once the pathogen is inactivated, it cannot move, for example, then macrophage can come and then engulf. Macam tu lah. Okay. So good, right? And then the next component, okay, the third component of the second line defense or the internal defense is the natural killer cell. Ingat internal defense, second line defense tadi kita tengok dia ada apa? Phagocytes yang pertama. Yang kedua adalah antimicrobial protein. Yang ketiga is this one is the natural killer cell, NK. Okay, natural killer cell. So the NK size of the NK is large. It is granular lymphocyte. It has granules. Okay, it has granule inside the NK, and it is produced or originates from the bone marrow. So the function of the NK is to destroy virus-infected body cell. The NK will not destroy directly the pathogen, but it destroys infected body cells, virus-infected body cell, and also tumor cells by both innate and antibody-mediated process. Okay, by both innate and also antibody-mediated process. Okay, so it says here, okay, the mechanism, okay, carried out by NK is that it releases a chemical molecule. We call it as perforin. Okay, perforin is released by the NK cell. Okay, perforin is a type of protein that forms pores. Okay, on the surface, on the membrane of the target cell. Target cell is the infected body cell. Okay, virus infected body cell. Okay, so the perforin released by the natural killer cell is inserted into the membrane, the plasma membrane of virus infected body cell to form pores. Okay, so once pores has been formed on the plasma membrane of the infected body cell, another molecule is released by the NK cell. So NK akan release two types of chemical, which is one perforin to form pores. Second one, once the pores has been formed, another chemical molecule, which is we call it as granzyme, another protein, okay, granzyme, will enter into the infected body cell and activate proteins inside the cell, which include apoptosis. Activate protein in the cell which include apoptosis. Apoptosis is natural cell death. It induces the cell to commit suicide macam tu, apoptosis. Okay. Okay, so kat sini, okay, so you have here the NK cell. NK cell, this is the target cell, infected body cell. Okay, infected body cell. So NK cell binds tightly to the target cell, and then what happened next is that the NK cell, in the NK cell, vesicles containing perforin, okay, protein that will form pores. Okay, macam antimicrobial protein tadilah, cuma antimicrobial protein yang complement system tu dia insert into the plasma membrane of pathogen, but this one it insert into the plasma membrane of infected body cell, okay, perforin, and form pores. And then the next molecule, which is granzyme, is inserted into the infected body cell, okay, release their content by exocytosis. Kalau you all tengok kat sini, this is the NK cell. Okay, so once the pores has been formed, and then kat sini you have the perforin forms, and then inside the NK cell you have another molecule which is granzyme. Granzyme will insert through the process of exocytosis. Exocytosis maksudnya initially the granzyme is inside the vesicle, inside the NK, kan? So vesicle will fuse with the plasma membrane of the NK through the process of exocytosis. Okay, exocytosis. So exocytosis releases the granzyme into the pores. Okay, into the pores of the infected cell. The pores is made up of perforin. Okay, the pores is made up of perforin from the NK. Okay, inilah NK. Release perforin, form pores, and then baru granzyme boleh masuk. Okay, granzyme boleh masuk. So perforin molecule polymerize in the plasma membrane of the target cell to form pores, and then granzyme passes through the pores and activate caspase enzyme that induces apoptosis in the target cell. Apoptosis is cell death of the infected body cell. Dia ada istilah, tak ada tulis kat sini. Saya ada pula. So you can Google the definition of apoptosis. If I'm not mistaken, is the cell undergoes cell death. Yes, cell suicide macam tu lah. Okay. So apoptotic cell is broken down into vesicle. Macrophage then phagocytose this vesicle. Okay, so the infected body cell akan break down into vesicles and then come along the macrophage. This will also exposes the pathogen. Okay. So and then the next component of the second line defense is the inflammatory response. Okay, inflammatory response. So it is non-specific response. Obviously, non-specific sebab dia adalah innate immunity. Walaupun second line defense is also non-specific for inflammatory. Okay, innate. Okay, so a non-specific response to infection or tissue injury. So minor injury or infection causes local inflammatory response, but more severe injury or infection may lead to systemic inflammatory response, which is fever. Boleh jadi demam lah. Kalau inflammation is throughout your body, you berasa panas seluruh badan, itu adalah demam lah. So demam tu adalah inflammatory response. Okay. So that's why response, so we call it as inflammatory response. It produces heat. Okay, heat. Alright. Okay, so local inflammatory response characterized by redness, kan? Kawasan yang awak luka, kan? Luka kat situ akan jadi warna merah lah, kan? So redness. Okay, and then warmth, and then swelling, edema, and also obviously pain. Okay, so you have a type of cell, we call it as mast cell. Okay, mast cell. Mast cell at infected or injured sites release histamine that have two function. Mast cell ni adalah daripada host cell yang ada pada injured site. Okay, injured site, the injured tissue will have mast cell. Inside the mast cell will have vesicles containing histamine. So histamine is released by the mast cell. Okay, so the function of this chemical molecule, okay, histamine released by the mast cell, is to cause vasodilation of nearby blood vessels. So at the injured site, you cut yourself, and then the mast cell in the surrounding injured tissue will release histamine. Histamine is the chemical or signaling molecule that cause nearby blood vessels to vasodilate. So when vasodilation occurs, more blood will flow to that injured site. So this causes redness. Lagi banyak darah mengalir kat kawasan yang luka ataupun infected area. So more blood flows due to vasodilation, due to the release of histamine. And then the second effects of histamine is that it causes the blood vessels to increase its permeability. Okay, permeability towards what? Towards, for example, tadi kita tengok kan yang the most abundant are the neutrophil. So this increases the permeability of the blood capillaries so that more neutrophil can penetrate from the capillaries and enters into the injured tissue, injured site, or the interstitial fluid surrounding the injured tissue. Okay, so it allows more fluid to move out of the blood capillary along together with the white blood cell, along together with the neutrophil, for example. So the neutrophil will then engulf any pathogen that tries to enter at the wounded area. Okay. Okay, so local inflammatory response, vasodilation of local blood vessel increases the blood flow to the injury site. Okay, so this causes apa? Redness tu tadilah, redness. Okay, red because more blood flows to the site, and also warm. Why warm? Because more metabolic heat is carried by the blood. So awak akan rasa macam kat situ ada panas sikit kan. So that is due to the metabolic heat bring by the more blood that flows to that injured site due to vasodilation tadi, due to histamine released by mast cell. Okay. Okay, so increased capillary permeability leads to the release of fluid, okay, blood clotting elements, and also phagocytes, monocyte, and also neutrophil to the injured site. So this causes edema sebab banyak fluid keluar kan daripada blood capillaries into the injured tissue and causes tissue swelling, edema. Okay, and also obviously pain sebab bila dah swell, the swelling causes pressure to be exerted on the nerve ends. Okay, due to edema. Kan? So pressure on the nerve endings due to edema causes you to feel pain. Okay, and then okay, so tadi increased permeability causes blood clotting element to enter into that wounded area, to injured site. Okay, to the infected area, for example. So blood clotting elements will wall off inflamed area to prevent or delay the pathogen or toxic from spreading into the surrounding tissue. So you have the blood clotting element formed so that this prevent the pathogen from entering deep into the tissue. Okay, so the neutrophil will carry out phagocytosis. It engulfs the pathogen that tries to enter at the wounded area or infected area. So neutrophils secrete signaling molecules that attracts monocyte, which will differentiate into macrophage to carry out phagocytosis. So neutrophil datang dulu sebab apa? Hasil dia terrilisnya histamine tadi by the mast cell. So bila neutrophil datang dengan banyaknya, lepas tu dia akan secrete another signaling molecule to attract monocytes. Okay, monocyte that will differentiate or matures into macrophage to carry out phagocytosis. Okay. Okay. So local inflammatory response. It says here activated neutrophil and macrophage releases cytokines. Okay, so this is the signaling molecules. Signaling molecules from neutrophil and also macrophage increases blood flow to the injury site, which leads to redness and warmth. So ada dua punca chemical molecule lah. Satu tadi histamine daripada mast cell. Second adalah cytokine daripada neutrophil and also macrophage. So these two chemical molecule will bring more blood to the injury site. Tapi histamine tadi yang akan menyebabkan vasodilation. Okay, vasodilation. Okay, so phagocytosis by phagocytic cell leads to the formation of pus, that is a mixture of that pathogen, tissue cell and also phagocytic cell. So formation of pus form that is a mixture of all these components, the pathogen, the tissues, okay, and also the phagocytic cell. Okay. So this is a diagram. Okay, so this is the diagram to show you local inflammatory response. So for example, you prick yourself with a splinter, cocok dengan pin ke, pin tudung, pakai tudung ke whatever, can obviously all the pin that you inserted into your skin, it is not clean. Okay, it is not clean. It has pathogen on the surface of it. Okay, so this causes mast cell. Okay, mast cell to release histamine. Okay, so this is the signaling molecule. Yang merah ni adalah histamine released by the mast cell. Okay, and then another signaling molecule also released by the macrophage and also by the neutrophil, which is the cytokine. Yang warna biru ni adalah cytokine. So cytokines from the macrophage, from the neutrophil will bring more blood to the injury site. Histamine causes vasodilation. Okay, vasodilation so that more blood will flow to the injury site. And also increase permeability so that more fluid can exit the blood capillary into the surrounding tissue, so that also more clotting factors, clotting elements can exit the blood capillaries into the injured tissue. Okay, so this is what happened first, the release of signaling molecules. Okay, histamine from mast cell, cytokine from neutrophil and also from the macrophage. Okay, next you have the antimicrobial peptide enters the tissue, neutrophil are recruited. Okay, so lagi banyak lah, lagi banyak neutrophil yang datang. Sebab apa? Neutrophil are the most abundant. Okay, most abundant. Okay, so when you are infected, you have a fever or you cut yourself, so yang paling awal akan datang dulu are the neutrophils. Okay, because they are the most abundant. So neutrophil, the macrophage, they are phagocytes that will engulf the pathogen that tries to enter at the wounded area. Okay, so neutrophil digest pathogen and cell debris and the tissue heals. Okay, so yang warm tadi tu kan, so dia akan cause ada redness and also warm. Okay. Okay, so next is the systemic inflammatory response. Tadi at specific area, you cut yourself, for example, or the area is infected by certain pathogen. Okay, so that leads to the formation of pus and all that, the redness, edema, semua tu. Tapi kalau seluruh badan, okay, is systemic inflammatory response. Okay, so toxin released by pathogen and pyrogen produced by macrophage lead to an increase in body temperature greater than 37.2, okay, causing fever. If your body temperature is above 37 degrees, so maksudnya suhu tu adalah suhu demam. Okay, so seluruh badan, tak adalah tangan dia rasa demam ataupun kaki je rasa yang panas kan. So it is the whole body. Okay, systemic inflammatory response. Okay, so benefit of fever, it promotes phagocytosis by phagocytic cell. Okay, so promotes phagocytosis, okay, by phagocytic cell. And then next is inhibit the growth of some pathogen by stimulating the liver and spleen to store iron so that blood iron level is reduced. Why is it necessary? During this time, during fever, for example, when your body is fighting an infection, the liver stores iron. Okay, the liver will reduce the amount of iron in the blood, because kalau kita tengok dulu masa chapter enzyme, okay, enzyme, in order for a living cell to work, it depends on enzymatic activity. Termasuklah bakteria ke virus kan. So in bacteria, in virus, in whatever pathogen, they also have enzyme that works inside the bacterial body or the virus body or the pathogen body. So this enzyme requires cofactors. Okay, cofactors. So kita belajar cofactor, coenzyme semua tu kan. So one of the cofactors are the presence of ions. So this ion, okay, will help in the activity of the enzyme. So when the amount of iron is dropped in the blood, so this limits the enzymatic activity of the pathogen. Okay, so that is why the level of iron is reduced during this time, okay, to inhibit the growth of pathogen. Alright, so and then accelerate tissue repair by speeding up chemical reaction. So that's why yang penting yang tadi warm tu tadi kan, so it speeds up the chemical reaction. Okay, so accelerate tissue repair. Okay, so far you have any question before we move on to the third line of defense? Faham ke? Kalau faham kita boleh proceed.
Faham, Madam.
Okay, so saya bagi seminit 2-3 minit untuk you all go through 4 komponen tu tadi dulu before we go to the third line defense. Phagocytic cell, antimicrobial protein, interferon and also complement system, NK, and also inflammatory response. Okay, so let's continue on the third line defense. Okay, so third line of defense, component of third line defense. Okay, so third line defense is also known as adaptive immune response or acquired immunity. Okay, acquired immunity. You gain that immunity. Okay, and then it is a specific defense. So this is the characteristics of third line defense. It is specific. Why is it specific? Because it specifically attacks specific pathogen. Okay, it's also known as third line defense. They attack very specific pathogen. Okay, using a small groups of receptor protein. So this receptor protein can be receptor protein is for example tadi kan yang kita cerita pasal helper T cell kan. So the helper T cell will have receptor, and so this receptor will bind to the antigen of the pathogen. Okay, so this antigen of the pathogen is specific for that pathogen only. Okay, so that's why it is very specific. So the response is slower. Kan, sebab apa? Nanti macrophage nak kena datang, engulf, jadi APC, lepas baru helper T cell datang to bind, and then helper T cell release cytokines, activate B cell, T cell. So cerita dia panjang. Okay, so that's why the response is slower compared to the innate immunity, where the response is rapid. Okay, innate immunity rapid, adaptive immunity slower. Okay, so it's activated after innate immunity takes effects. Kan? So innate immunity yang tadi kita tengok ada second line defense kan. Second line defense melibatkan macrophage. So ada overlap kat situ, macrophage in the second line defense, engulf, jadi APC in the third line defense. Macam tu. Okay. So found in invertebrates. Okay. Found in invertebrates. Yeah. So two types. You have the antibody-mediated immunity or the humoral immune response. So this involves the B cell producing antibody that circulate in your body fluid. Humoral, it refers to body humor. Body humor is your body fluid. Your body fluid is blood, lymph, and also interstitial fluid. Okay, and then cell-mediated immunity. Okay, so cell-mediated immunity, this one involves the cytotoxic T cell. Okay, anyhow, these two antibodies mediated immunity and also cell-mediated immune response, it requires the formation of APC and also the binding of helper T cell ataupun tadi T helper cell, samalah kan. Guna yang barulah T helper cell. Okay, T dulu, helper cell. Okay. So adaptive immune response, the antibody-mediated immunity or the humoral immune response involve the activation and clonal selection of B cell which secretes antibody. Okay, antibody is produced and secreted by the B cell in the humoral immune response. Okay, so nanti kita tengok what is clonal selection. Okay, what is clonal selection? Sama juga for cell-mediated immunity. Okay, so involves the activation and clonal selection of T cytotoxic cell. So dalam buku rujukan yang you all guna, yang bukan Campbell, dia letak T dulu. Okay, T helper cell, T cytotoxic cell. Okay, so that recognize and destroys the pathogen. Okay. Okay, so major characteristics of adaptive immune response. Okay, so characteristics of third line defense. So this one you have to know. Okay, what are the characteristics of adaptive immune response? Okay, first tengok rapid response to a second exposure of the same antigen. Okay, rapid response kalau awak kena infection of the same virus for the second and subsequent time. Okay, tapi kalau respon dia lambat, slower for the first infection. When your body is infected by a certain virus for the first time, the response is slower. But when you are infected by the same virus again in the future, so the response is rapid. Kenapa? Nanti kita akan tengok. Basically, dia melibatkan penghasilan memory cell. Okay, memory cell that are stored in the lymph nodes. Okay, that are stored in the lymph nodes. So that's why they rapid. First time exposure, tak terhasil lagi memory cell. Tapi second and subsequent times of exposure, your body has already produced the memory cell. So tak perlu nak undergo engulfing, APC, T cell, helper T cell, semua tu tak perlu. Dia terus pergi kepada memory cell. So the presence of the memory cell in the lymph nodes causes your body to react faster against the infection by the same pathogen later in the future. Okay. So the second characteristics of adaptive immunity is that it detects antigen never before encountered. Okay, so whatever antigen from a pathogen, a new antigen or a new pathogen that enters into your body, will be detected by specific receptor of your white blood cells. Okay, white blood cells of helper T cell or B cell. Okay, So and then detect antigen ni sebab apa? Akan ada inside your body there are so many white blood cells that have many different types of antigen binding site. Okay, many different types of antigen binding site. Okay, on the surface of the lymphocyte. Okay, of the T helper cell akan ada banyak reseptor. Sama juga dekat on the surface of B cell akan ada banyak different types of receptor. So this receptor, when it binds to the matching antigen, so the receptor, the B cell or the T cell with the matching receptor will be activated and undergo clonal selection. Okay. So another characteristics of adaptive immunity is that your immune system should know the difference between self and non-self antigen. Okay, self antigen and non-self antigen. So self antigen are antigen on the surface, okay, of the host cell. Self antigen adalah kepunyaan host. So the host cell should not attack the immune cell of the host should not attack self antigen. Dia tak boleh serang awak punya sel badan sendiri, sebab that will lead to autoimmune disease. Tak boleh. Okay, so it should only attack non-self antigen. Okay, non-self antigen is the antigen on pathogen, for example, the antigen on the bacteria, the antigen on the parasites, okay, whatever that are foreign to your body, okay, that is non-self antigen. So your immune cell should only attack non-self antigen, cannot attack self antigen. Okay, so next is the cell proliferation. Kan? What is cell proliferation? Once the specific B cell or T cell with the matching binding site to the antigen is activated, so this cell will divide into many cells, many cells. We call it as clonal selection. That leads to cell proliferation. Dia membahagi dengan banyak. Okay, cell proliferation, proliferate, membahagi dengan banyak. Okay, cell proliferation increases the number of B and T cells specific for that antigen of the pathogen. Okay, specific for that antigen or pathogen. Okay, for example lah, dalam badan awak ni, saya bagi 4 lah. B cell 1, B cell 2, B cell 3, B cell 4. Each of these B cell will have receptor with different antigen binding site. Whatever lah kan, macam tu lah. Okay, so ini macam tu lah. Okay, so you have, ini 4 saya bagi, tapi ada banyak lagi dalam badan awak. So 4. So these 4 different B cells will have different receptor on the surface of the plasma membrane with different antigen binding site. Jadi kita namakan dia sebagai antigen binding site. Okay. At one time, when your body is infected by certain virus tu dekat surface antigen dia, surface cell dia, shape of the antigen is triangle. B cell mana will be activated? Which B cell will be activated?
B1.
Okay. Okay, B1. So the antigen of the pathogen will bind to the B cell. So this causes the B cell to be activated and undergo clonal selection. Clonal selection maksudnya dia hanya select B1 saja. That is clonal selection. B1 saja. Yang lain B2, B3, B4, whatever will not be activated. Only the B cell with the receptor matching to the antigen will be activated and undergo clonal selection. Once undergo clonal selection, this B cell will proliferate into many cell. Okay, into many cell. So B cell yang ini asalnya ada satu je yang bind, so dia akan jadi banyak. Okay, proliferate. So the proliferation of this B1 cell ni, contohnya, dia akan jadi kepada dua koloni yang berbeza. Satu, kita namakan dia the activated B cell. Activated B cell is the plasma cell. Okay, activated cell lah, which is plasma cell. The activated B cell or the plasma cell is the one that produces the antibody that circulate in your body fluid. Another set of clone is the memory cell. Okay, the memory B cell. So this memory B cell will be stored in your lymph nodes. Okay, so jangka hayat memory cell ni adalah lama, puluh tahun. Okay, so when you are infected again by the same virus, by the same pathogen, your body is ready to be protected because your body has the memory cell for that specific pathogen. Sama juga kalau dia melibatkan T cell kan, sama jugalah kalau cytotoxic T cell, sama juga. Okay, the same mechanism. So once a certain cytotoxic cell is activated, it undergo clonal selection, proliferation, hasilkan kepada dua group. Satu the activated cell, satu is the memory cell. Okay, so cell proliferation increases the number of B and T cells specific for that antigen. Okay, so next is cells involved in adaptive immune response. Okay, so there are two key cell types, which is lymphocyte and antigen presenting cell. Okay, sekarang ni dah pukul 9.50. Saya rasa kita stop dulu sebab dah banyak dah information given today. Saya tak nak bagi awak tepu sangat. Okay, so just kita tengok dulu, kita stop dulu sampai yang characteristics of adaptive immunity for you to understand and digest for our next lecture, where we are going to look at the detailed mechanism of the adaptive immune response. Saya stop dulu sebab dia punya mechanism dia panjang. Okay, so kita stop dulu until our next lecture, which is next week. Kita kekurangan 1 jam sebab hari tu hari Selasa cuti. Firstly kita boleh cover, okay, tapi tak apa. We try to cover next week. Okay,